Yeah. Okay. So let's first introduce us. We are a small group of research students, mostly from IIT BHU Varanasi, and going to form a Sigma Chi student chapter here. Sigma Chi is a non-profit scientific research honor society for scientists and engineers at in the international level. And we organize scientific discussion sessions with experts from multidisciplinary research area. Given this, for the February session, we have requested Dr. Mukund sir to deliver a lecture on his fantastic works on entrepreneurship and the research, how to connect the academia and entrepreneurship. So now to introduce our speaker, Mukund sir is a serial entrepreneur and founder, president and chief scientific officer of Think Pharma and Ayurveda Healthcare Innovations. He earned his bachelor's and master's degree from University of Pune, India. Later on, a PhD in organic chemistry from Georgetown University. He is having years of scientific experience from academia to industry, and also a recipient of 3 Scientist of the Year awards. His research interests are in drug discovery and development, process chemistry, derived medicinal chemistry, traditional Indian and Chinese medicine. So I will not take much time. He will explain in detail about his work in this talk. So I must thank Dr. Mukund sir for accepting our invitation. Please proceed for your talk.
Well, thank you very much both for the invitation and for this extraordinarily kind invitation to address you. This is something that is very near and dear to my heart, reaching out to a broad audience, hopefully getting them excited about chemistry, about the organization like Sigma Xi, and actually stimulating participation. It's indeed a privilege that I take very seriously, and here we go. So my last name, Chorghade, by the way, my family hails from Nagpur, and the name Chor comes about in a very interesting way. My great whatever ancestor in the year 1773 won a battle for the Maratha Peshwas by stealing the horses of the opposition. So those horses were stolen from the Nizam of Hyderabad, and that enabled the Maratha army to win, and therefore the family was given the name of Chorghade.
That's a wonderful story.
I'm not here to steal any horses, but I'm here to steal your hearts. So what mischief do I create? The name of my company is Think Pharma, which obviously has pharmaceutical connotations. And the T is for technology, then health, innovation, novelty. Why did I change the K to the Q? Well, because all the pharmaceutical data has to be generated in the quality sections. And I have a childlike fascination for being the Mr. Q of James Bond fame, trying to discover new technologies and all that. Over the years, of course, I've had a career in both academia as well as in industry. I got my postdocs, as it was said in the introduction. Then I had an industrial career that spanned Dow Chemicals, then Abbott Laboratories, Cytomed, and my last industrial position was as a vice president of chemical development at Genzyme. After all the management restructurings and all that, I decided I'm going to become an entrepreneur. But I set up my companies, but I also retained a foot in academia. And today I am very happy that I am an adjunct research professor at some very good institutions, where one of the joys of my life is to explain some of the perspectives to the young students and all who are going to be our pillars of tomorrow. Now, when people ask me, Dr. Chorghade, how come you keep a foot in both doors, in industry as well as in academia? My answer has been the same. Whenever I'm in an academic group and I may talk something I shouldn't, talk some nonsense, I have a ready-made escape route, because then I can say, I don't know, I'm from industry. And the same thing works in reverse as well. Over the years, I have become a big fan of innovation. Most of us do a lot of research that is inventive. We file patents, we do this. But what is innovation? It is going to be the art and science of literally making revolutionary changes in thinking. Innovation is not the same as jugaad. Now in India, sometimes people use the term innovation, oh, kuch bhi kar lenge, and that is not indeed going to be a path forward. India is full of some extraordinarily creative people. The name Banaras Hindu University was mentioned, and of course, that is hallowed turf for many people. The central lesson I want to convey is that invention is the conversion of cash into ideas. All of you generate cash through grants and everything, through scholarships, and then you go into the lab, you make an inventive contribution, and then you generate more ideas. Innovation is the conversion of ideas into cash, because innovation theoretically and literally means that you have to get your idea, your product out into the market where maximum number of people can be benefited from it, maximum number of people can use it. Nikola Tesla was an inventor, spent a lot of money to create inventions, but was never able to monetize it. I live in New Jersey. Thomas Alva Edison's lab is about 20 plus miles from my house. And look at him. He was not exactly a college graduate, but he had 1,000 patents. He knew how to make money from his ideas. He set up companies which hired over 60,000 people. Take a look at the sewing machine, which was invented by Elias Howe, but it was commercialized by Singer. And even my grandmother had a Singer machine everywhere, and it became almost a legendary byword. Oh, let me just use a Singer machine to do that. Similarly, J. Murray Spangler invented the electric suction sweeper, but it was W.H. Hoover who knew how to market and sell the vacuum cleaner. And such stories are indeed legendary. Please keep in mind, and one of the things Sigma's Eye could be very instrumental in is to make sure that our scientists contribute a lot to the innovative landscape. Invention may occur in a laboratory, but innovation simply must take place in the marketplace. It is a creative process that must include all these business functions to be very successful. Now, I just told you that all my career has been in pharma. Now, when I came to the United States, of course, the watchword was, the USA has the best universities in the world. They have access to the best students in the world, best facilities and all that. So therefore, we are ideally set to contribute to any enterprise, but particularly discover new drugs of benefit to mankind. By and large, we succeeded, but the side effect of that was something very, very different. We got to be the largest drug consuming country in the world. Take a look at what this famous cartoon says, that we become addicted to drugs at the age of 0 to 4. We start taking amoxicillin. Then the parents take their kids to the doctor. Oh, my kid wants to get into Harvard. So doc, please prescribe him some ADHD drug like Ritalin. Then we go to high school. We take appetite suppressants because we want to look good. Then we enter college. We take no-dose tablets to party late or study late. Then we balance the pressures of family and career with Prozac. Then you reach my age group. It used to be called old, but now it is called chronologically gifted. I really prefer that. And above 65, many elderly Americans in particular are taking between 12 to 19 medications per day. And that cannot be good for you. So I said, well, how can I realistically contribute? My postdoctoral work, of course, I synthesize anti-cancer drugs. I was involved in the total synthesis of a fascinating natural product called as bleomycin. In those days, all the natural products was done by isolation, structure proof, and then synthesis to prove its point. And palytoxin, my postdoctoral work at Harvard was with a gentleman called Professor Yoshito Kishi. Unfortunately, he just passed away 2 weeks ago, and I owe a lot to him. So therefore, this drug is molecule is the most complicated molecule that has been synthesized. It has 64 chiral centers and therefore more than 1 trillion possible isomers. Now, remember what I said, that in those days, the natural products used to be what? Harvested. Traveling salesmen would come to India, China, and all that. They will scrape up the soil samples from their shoes even, and then subject them to analysis. They would take away plants, and they would prefer to do this research in their own laboratory. Many of the natural products got developed into drugs, and even in the early days, I'm talking of hundreds of years ago, natural products were the only effective medicines known. Even medieval monasteries had herb gardens and so on. And look at what happened in China. So there was a scientist called Yu Tu. She looked at papers from parchment papers, even from 168 in the CE era, 200 AD, etc. And then she analyzed all these samples and was very successful in discovering a drug which is called as artemisinin. And of course, she was rewarded with a Nobel Prize. This was the first example of somebody going back in time, analyzing the data, what was written up by the wisdom of the sages, and therefore isolating a drug for it. Now, here are some fascinating stories that I started reading. All this work was suggested to me by a legend in the field. He's a good friend of Yu Tu's. His name is Dr. Ashok Vaidya, lives in Bombay. He has an MD, PhD from Yale and Columbia. So does his wife. Then when the Ciba-Geigy Center closed down, he decided, why not? He studied this Indian field of Ayurveda. And this is what he got me into. So take a look at how some of our fundamental drugs were discovered years ago. Many villagers would suffer from convulsions, fever, particularly at night. We now know of this as malaria. They used to go and get water from the watering holes in the area, and then they will be cured. Their fever would be reduced and so on. Then the villagers made an observation that lions, tigers, leopards, pumas, all of them would suffer from the same problems. And it is these animals that will also be cured. Now go further down the years, as down memory lane. And many of the people then found out that around these watering holes, there were trees. They were the cinchona alkaloids. The leaves from the trees would fall in the water. And now, of course, we know of these molecules as quinine and so on and so forth. So Pelletier and Cavanto did the structure proof, and they isolated this drug. And this story goes on. Legend had it that King Charles I was cured of malaria by consuming quinine, and then Friedrich Serturner isolated morphine from opium or poppy seeds. All this was done from Indian natural products, and then people isolated further the codeine. Of course, poppy seeds are a big source of what? Of essentially some things like opium, which you should not be consuming. But the chemistry was done and well documented. Today in the world, there are many natural products we all use as medicine. This is a plant called as the periwinkle, which is Cantharis roseus. This was the prime source of vincristine and vinblastine. Today in India, this grows ubiquitously in so many backyards. But people do not bother to take this to prevent cancer or something like that. People prefer to buy the very expensive medications obtained by isolation. And then, of course, you're familiar with camptothecin and so on. Now, over the years, people have always wondered, are you discovering medicines or poisons? But we have discovered many medicines. We got cocaine from the coca tree, tubocorarin from here, pilocarpine from jaborandi tree, bromelain, and so on and so forth. So we had always the knowledge. Now came a challenge of merging it or marrying it with the chemistry. Now, when I was a young man, of course, natural products in the West started declining because many people branched out into the non-traditional natural products. They said, well, we'll define natural products as arising from bacteria, mollusks, and fungi, and everything else. People would go deep sea diving in Hawaii and then isolate some natural product from some sea animal. It was intellectually a tremendous effort. It was wonderful, but the molecules were so complex, you could never subject it to total synthesis for commercialization. So your commercial availability was very little. And then all the countries started claiming these products as their own natural product, intellectual property. And then we had things like the Phuket Declaration and so on, which said that every natural product must lead to some intellectual property for our people. Then we had some boondoggles. We had the boondoggles of combinatorial chemistry that was met with extremely limited success, and there was no real knowledge transfer mechanism. Your scaffolds were not diverse, they had no chirality, and therefore it simply led to a commoditization of art and science. The newer biology was retrofitted to very mundane chemistry, and India and China were looked upon merely as a low-cost alternative. I have always objected to this one. India is not there, China is not there to make things on the cheap. It is there to harness the brilliant intellectual power of our people. Now, I would like to explain some of these things. I have always had the highest respect for the intelligence of people from India. We are innovative at heart. There is a famous story that appeared that a young man went to the Chemical Bank of New York, asked for a loan of $10,000. Well, in response, they said, what will you pledge as a collateral? He said, I have got my 560 Mercedes car, SE. They said, that's a $80,000 plus car, but they gave him the loan of 10,000 at 9%. They took his car keys and took away the car in the basement. 3 weeks later, he repaid the loan, paid the interest, 26.72. The manager asked him, we don't understand you Indians. What are you up to? You know, you had $500,000 in your saving checking account. So why did you need $10,000? He said, this is our innovative mindset, my friend. I was going on a 3-week business trip to India. To park this expensive car in Manhattan would have cost me $48 to $50 per day. I've got the same job done for you for 3 weeks at 26.72. Now, doesn't this tell you a lot about where creative and innovative thinking can come? So we in India, and the gentlemen who are setting up the chapter in Banaras, you have all the intellect, you have the wives, BHU is of course over there, and the India can be a gateway to international collaboration. Now, in this field of Ayurveda, Sir Richard Roberts, who has been a collaborator of mine, he came up with a beautiful statement. If you do not take food as medicine, you will be soon taking medicine as food. And too many people are addicted to too many medications. But take a look at what our ancient wisdom has taught us. Take a look at the simple Ayurvedic thali that we have. And you know, this is shown in many colorful forms. In the South India, particularly, it is used on banana leaves. But we always have raw onion. Even if you go to a simple bar and order something here, they will serve the raw onion. That serves a tremendous purpose because in all the traditional wisdom, it has been proven that this contains ingredients to ward off sunstroke, heat stroke, nosebleeds, and everything else. Now, Professor Eric Bloch in SUNY Albany has extracted this and sold it in America as anti-nosebleed remedy. We have not done adequate amount of research. And you keep going on all this. Every one of these ingredients, what we call sattvic food, it really means something. Now, my grandmother, my mother, they used to give me pomegranate seeds on my breakfast yogurt. That contains alkaloids which are known to ward off diarrhea and dysentery. Did they know the chemistry? No, they did not. Now, 12 years ago, Harvard published a paper says if you drink 2 glasses of pomegranate juice per day, it will regress prostate cancer and it will have a beneficial effect on breast cancer. Think of the implications of that. This is our traditional knowledge, know-how coming to the fore, and we owe it to ourselves to globalize this, to monetize it. If necessity is the mother of invention, surely frustration is the mother of innovation. And today I'm making a call for innovation to be the new normal. So what is this Ayush therapeutics? It's a combination of Ayurveda, yoga, Unani, Siddha, and homeopathy. Now, yoga and homeopathy are not in the traditional realm of chemistry and molecular biology. So sometimes people use the terminology of Oz. Now, in Ayurveda, which is a tremendous discovery engine, there are 300 well-documented and in-house medicinal plants. sound philosophy, 5,000 traditional formulation. Let us focus our efforts on doing some work. Can I do work on 5,000 formulations? Well, heavens no. Let us focus on, say, the top 10 medicinal plant. So this is what my current effort is. This is what Think Pharma and my company Ayurvedya does. I'm looking for volunteers for the cause. So now our path is well defined. We take plant extracts, we study the in vivo activity, and then this path can be very nicely lined up. We do a limited amount of clinical trial as well. Now, what happens with Ashok Vaidya's thing on observational therapeutics? Today in India, if a person, man or woman, goes to an Ayurvedic physician, in what language is the dialogue conducted? Any one of 26 plus Indian languages, any one of 800 dialects. That is not always written down. So we need a very cohesive framework to analyze the extracts that are being given, how the patients are being treated, and so on. I recently found a paper. So what Vaidya wants to do is to take this experiential data and bring it back to the lab. So it is a bedside to the bench, which turns the traditional thinking on its head. Now, I read a paper on the hybrid molecules where people coupled artemisinin, chloroquine with vitamin K3, two anti-malarials coupled here, and they got something effective in oncology. Can we take this technology further? And I found my company on the basis of that. I, of course, made some interesting compounds where we had two scaffolds all integrated into a single molecule. So this particular one had a quinolyl chalcone, and we achieved a lot of success. What kind of basic information we have? I told you, let's take the top 10 medicinal plants. Now, for example, Soralia cordifolia, this is called as Baguchi. These are the ancient Sanskrit that was written up by, again, all the wisdom. But people have now trying to exploit it for immunomodulatory effects and so on. Gingiber officinale, which is called as Shunthi, acts on certain biological targets. We now know the chemistry. Can we not bring the transforming power of chemistry? Can we not bring the transforming power of molecular biology into this one? Now, an organization like Sigma Xi, it has a collection of scholars in physics, chemistry, molecular biology, and all. Can we not focus and think of a way of monetizing all this traditional knowledge? But what Sigma Xi also has is ethics. And we have to conduct all this work with the maximum ethical ways possible. So let us not take any shortcuts. Let us treat the patient with the due respect that these patients actually deserve. And sometimes that is a lesson that has been a bit lost. So what am I trying to create? My company is focused on aligning yesterday, today, and tomorrow with traditional medicine and cutting edge science for discovering these medicines of tomorrow. Now, what is the conventional approach of Big Pharma? Big Pharma has always wanted the blockbuster model, and they want one drug, one target. So you have to establish the human proof of concept, etc., etc. Now, you start with the animal models. Many of them, they are not necessarily effective, are they? But the noted scientist Joshua Friedman in Harvard Medical has said, I have cured cancer in laboratory rats hundreds of times. I've never yet done it in a human being. That tells you something. What is our approach? We are following up on Ashok Vaidya's lead. In Ayurveda, there is one drug, multiple targets. So therefore, we can use some of this knowledge, some of these chemical scaffolds, some of the molecular biology to target more than one disease. Why will it be low risk? It will be because people have been, they are not even patients at the time, they have been using these for centuries. And let's take a look at, so this is the definition of reverse pharmacology. It is a science of integrating all these bedside documented experiential hits into leads. We, of course, conduct the exploratory studies. And this is the basic philosophy that we will be able to shorten the duration of clinical trials. by several years. Why? Because people have been taking, nobody has died from eating one more pomegranate or two more pomegranates or everything else. The toxicity and the dose levels can be controlled. And that is ultimately, we have a whole series of compounds. There are 10,000 plants, but we have succeeded in isolating many of these molecules. You know, this is curcumin, gingiferone, and so on and so forth, which can be very valuable scaffold. Now, much of pharma has not exploited this because in the traditional combinatorial chemistry, you use robots, you use the Heck reaction, and you got some very flat and chiral molecules. These are some of my favorites, which we have worked on in mangiferin, of course, coming from mango, and then we can put together all this. There's a plant in India called as Stenospora cordifolia, and there has been a tremendous amount of work published by a woman I never had the pleasure of meet, Sharadini Dahanukar, who passed away in 2002. But she has published many papers on this particular field. Now, take a look at what is going to happen. Many of these molecules have been used to reduce your blood sugar. They have been useful for treatment of diabetes. We have things like bitter gourd, which is there in every Ayurvedic thali. That also reduces your blood sugar. What do most people do? Array, I have had my daily dose of bitter gourd and guduchi. So now my blood sugar is going to be low. So I'm going to be drinking 2 cups of heavily sweetened tea, and I will have 6 gulab jamuns, which also carry a lot of sugar. That is not the way of ensuring good health care. Let's look at this. Now I came to the observation. Remember, some years ago, the American diabetic drugs, they were all going down because they had cardiovascular risk. Then we realized that Mother Nature has made these leaves heart-shaped. The people told me that when people with a heart attack are being taken to hospital, they are fed a little bit of the extract of Stenospora cordifolia. That revives the heart. It's a stimulant. Can I not make a hybrid of these molecules derived from these with the anti-diabetic drugs like Avandia, like pioglitazone, and so on? And we got some pretty interesting results where the cardiovascular risk could be ameliorated. Now we are working on the problems of scale-up, but these are some fabulous molecules like berberine and so on and so forth. Here is a fascinating story. This came to us from South Africa. Many of the natives who live in Africa, when they're famished, hungry, far from home, tired, and they're waiting for a bus, they are hungry, they munch on the glabrous roots of this plant called as Hoodia gordona. And then they have an instant feeling that their hunger pangs have subsided. They have a feeling of satiety. Now, Pfizer was willing to commit $2 billion for development of this for treatment of obesity. We in America do not work on treatment of obesity very well. Most Americans love to go to a McDonald's, order a large triple cheeseburger, supersize it with French fries, large apple pie. Oh, by the way, give me a Diet Coke. Then they feel that, okay, their instincts are quite satisfied. Now, this plant only grows 6 inches in 12 years. So it is not all that sustainable from a global perspective. But we found some sophisticated computer analysis, and we in Maharashtra, we in Kerala, and in South India, we have these plants which are called as Caralluma fimbriata, which act by the very same mechanism. Okay, so therefore, they contain the same sugars, they contain very similar steroidal aglycones, and we can do it. In our research, we led to this biologically active macrolide, all derived from reverse pharmacology. New 10-membered macrolides, these were isolated for use against malaria, all derived from indigenous Indian plants. Decarestrictin lowers your cholesterol. This molecule lowers your blood sugar. So all of these have potential uses. Where do I use the chemistry? I said, remember, we have to develop chemical processes which will make these very scalable. So we set ourselves up to this task, and we quickly realized that this ring is in fact a large ring lactone, which it will be difficult to synthesize. But we did that. We assembled this, and then Professor Bob Grubbs, the late Nobel laureate, who was a good friend of mine, he simply suggested and that he will give us the catalyst, we would deprotect, and then we would cyclize. We got a remarkable stereospecificity, and then Dondi Ramesh, my graduate student, did the reaction in reverse and got the other enantiomer as well. We could refine this synthesis with desmethylnoninolide and get exclusively the E isomer. So you can see where we are indeed going with this one. We are using the transforming power of chemistry to really do something good. Here are some couple of fun examples. So Daucus carota, this ketone was reduced with Daucus carota to a mixture of alcohols. Now you will say, what in God's name is Daucus carota? It is carrots. We demonstrated this reaction on a pharmaceutical intermediate. We pureed the material with carrots in a blender, let it sit, and of course we got this. Then the pharma company says, can you make us 200 kilos of reduction? We calculated it will take us 4 tons of carrots. We said, no problem. We sent our lab people out to the market to buy 4 tons of carrots, which we will then put into the reactor. Within the hour, we were visited by the local police who thought we were bad guys. Next day was a famous festival in India where everybody wanted to make gajar halwa at home. And therefore, they thought we are bad guys trying to run up the price. Well, we persuaded them that that is not the case. This is another famous story of a molecule that was useful for psoriasis, also for a disease called as thalassemia. One of the raw materials was cysteine. I got a call from Novartis that this material is $240 a kilo, and we in India were selling it for $3 to $4 a kilo. Are we that good in process chemistry? The address of the plant rang a bell in me. It said near Tirupati temple complex. You know that everybody goes there, ceremoniously donates some of their hair. Some entrepreneur found out that you can indeed collect these hair and then hydrolyze them to cysteine. When this word reached America, the US FDA said, oh, we want to see this facility. I said, oh, wait a minute. This is not a big GMP approved facility. People go in, there are a lot of barbers sitting there, snip, snip, and then somebody ladles this into a big vat. They said, no, we have to see it. The day the FDA showed up, there were other 3 people who were there who, by their dress, it was very clear that they are Orthodox Hasidic Jews. So I got to talking with them. They said, do you realize, doctor, that that product, cysteine, also goes into kosher foods. It also goes into making of wigs for women. So we as Orthodox Jews and the rabbis representing them, we have to certify that this is a kosher process. So on one day, we got a positive rating from both the US FDA and we got a rating from the Orthodox Jewish community as well, which added some fun and to my knowledge and everything else. Now today, what are going to be the trends? What are we involved in? I'm very closely affiliated with University of Illinois, where a lead scientist called as Martin Burke does all this particular work. And what does Martin Burke say? That we have invented so many natural products. Now he has analyzed them. All of them can be into a cluster of 1200 to 1400 scaffolds. Now think about it. Can we not build, fold, assemble, make many unnatural products by coupling of these into various permutations? This can be a moonshot for chemistry. It is like the natural productome project. So we have to define a Lego set for these natural products, which has been duly done. And this is Marty Burke's proud invention. It's a small molecule synthesizer, which is getting him all kinds of attention. They have set up these maker labs at Illinois. And if I may, I would invite him to give one of the Sigma Xi talks. Now, they always say, Dr. Chorghade, what is your proudest contribution to research? Yes, I have done process development of drugs and all. My proudest contribution came when I was a medicinal chemist. And I started seeing that the compounds that were made were slowly administered to rats, dogs, and everything else. Then the animals were sacrificed. Then you analyze their bodily fluids like blood, urine, sweat, and everything else, saliva, for metabolites, which were generated in pitifully small quantities. The metabolites were hardly standardized. The observed drug metabolism may not be consistent, and we started getting a lot of pressure from the animal activists who simply did not care for the fact that we were sacrificing so many animals. You all do recall that the government of India banned the export of rhesus monkeys several years ago. And so, what was my contribution? I said, I'm a chemist. So, can I not define a chemical catalyst which I can weigh accurately, which I can mimic all this drug metabolism, provide a very rapid and effective means for predicting the interactions and toxicity of xenobiotics? In doing so, I will be reducing the amount of animal sacrifice. Now, Mother Nature is the greatest synthetic chemist of all time. She has invented enzymes called as cytochrome P450 enzymes. So these are sterically protected and electronically activated. She has a proteinous sheath to doing that. Then she does this one electron oxidation, and anytime we get drug, they are converted to metabolites. Our technology works by similar mechanisms. We are creating a chemical catalyst. Now take a look at the early metalloporphyrins. Emil Fischer got a Nobel Prize on it. They all have a tetra-asa macrocyclic structure. But the problem is, this is an incredibly flat molecule. And as it tumbles in solution and all that, it simply gives the mu oxodimer. And that limits its industrial use. The turnover is very low. The molecule pretty much is useless. At minus 80 degrees, it has a turnover number of 20. Now, when I was a young man, when my professor would ask me why something doesn't react or why something reacts fast, and I had no idea about the answer, I would raise my hand and say steric hindrance. And by and large, I could be correct. So I decided to use the same steric hindrance, build in some picket fences around the molecule, put chlorine atoms, then the molecule will be puckered, saddle shaped, and it will not dimerize. So we generated all these catalysts by incorporation of halogen atoms into the asa macrocycle. We put sulfonate atoms, and we could mimic many of the reactions which our human liver does. So we can take the O-methoxy group, particularly found in morphine and all that. We could generate all these metabolites. We can convert all the way to the phenol. Now, who is asking for these? Remember, many people who are drug addicts, basically, they overdose on morphine, codeine. When the authorities show up, they have no standard amount of metabolite sample. They cannot analyze the blood because these people are dead frequently in half an hour. There was a famous actor called Hoffman who took in a syringe a mixture of crack, cocaine, fentanyl, and all that. And therefore, the authorities sometimes lack it. But in the Justice Project, we have got some grants for doing this. We can catalyze all the reactions of drug metabolism. We can do phenols, we can do phenones, and so on. And then I decided to subject it to the test. We could take a known drug called aminopyrine. As organic chemists, I could predict where the metabolism will go. It will be N-demethylation, etc. And lo and behold, we found exactly the products that were reported in the literature based on what the human liver acts on all these drugs. But we got incredibly lucky. We found 4 new products that were formed. Then we sent it to the innovators. They said, oh my God, these are the ones that are implicated in the toxic side effects. So we were making a good contribution to that. Then I was confronted with a problem. Here there was a double bond, and every computational chemist, every molecular biologist said that surely, Dr. Chorghade, this will form a very toxic metabolite which will be very detrimental to fetuses and to pregnant women because they had the thalidomide tragedy at heart. I said, okay, we'll try to isolate that metabolite. We made 18 grams. We proved it was toxic. Then we said, let's hydrogenate. You will get new metabolites. and therefore this drug went to the clinic and which got sold as odapipam. Now, I was listening to a tremendous lecture in Harvard University, and in that the people were saying that all the elderly people in particular take 12 to 19 medications per day. Again, I said, what about steric hindrance? If these drugs associate even in your gut and everything else as they are going down your GI tract, surely in the liver, they'll associate and form a larger molecule. Then your liver won't be able to act on it. It won't be able to participate in the normal ADME process, absorption, distribution, metabolism, and then excretion or elimination. What will happen if these large molecules form? You will have a large amounts of undissociated drug left in you. One doctor got up and said, so what? You'll be cured faster. No, you will not, because the therapeutic window is an extremely narrow one between toxicity and this. So we decided to study this, and we reasoned, let us take some drugs which will not associate. So this is our friend Lipitor, and we took this molecule which was developed at Bristol Myers. It is a highly sterically hindered adamantyl ring. This will not associate. So it will all go down separately. It will each metabolize by its own pathway. So you will not have the side effects. But drugs like this one, which was invented at Merck, it was anacetrapib, you have all these atoms now which can associate by hydrogen bonding. So this drug we predicted will literally get stuck in the human liver. And so it proved. This thing simply was so non-polar, it will get stuck in the fatty acid tissue. It gets stuck 6 weeks after the drug is discontinued. And that cannot be good for you. Now take a look at what did I tell you about animal sacrifice? We wanted to reduce it. So when I was lecturing in Germany, one very aggressive set of activists walked in and they said, Dr. Chorghade, why do you do all this rubbish anyway? And then they asked me, why don't you Americans work on lawyers instead of rats? And the reason was, her reasoning was that in America, you have more lawyers than rats. There are many things that lawyers do which even rats don't do. And many scientists will get more attached to the rats than they will to the lawyers. Now everybody was laughing. I decided the only way to save the day for us scientists was to say, but ma'am, any data that I get from lawyers is not going to be extrapolable to the human population. At that point, she laughed and she came and talked. Today, we are making enormous progress. We have defined this as the chemosynthetic liver. These catalysts are called as the Chorghade dolphin catalyst. But what is pleasing to me is I have on the website of PETA and the Anti-Vivisection Society as a right-thinking scientist who has the welfare of animals at heart. This kind of thing has pleased me enormously. In closing, let me give you a couple of examples of innovation, not by me. This is a scientist called Dr. Naji Abumrad. He was a doctor who treated a lot of athletes who were engaged in very strenuous medication. What were these people doing? They were doing strenuous exercise. They will pull their muscles and so on. And then they will be in a lot of pain. So this gentleman found out a metabolite of lysine, which was called as HMB. And that metabolite resulted in something which was the healing of the muscles. That became a drug on its own. Then they found out that above the age of 40, you lose half to 1% of your muscle mass each year. Why not give this to people in medicated drinking water? And now they are marketing this drug as this molecule in mineral water, which is sold as Nirvana in all the stores and so on. You can buy this at airports in the USA and New York. They have bought this land. There is 1 trillion gallons of water plus in this underground aquifer. Now here is some light-hearted humor. There was a scientist called Professor Gilbert Storr, Carl Gerasi, and Johnson. They added this as an additive for a contraceptive drug. Now the question was, why is this the ideal contraceptive? Because it says no in every position. Okay, this is hexanitrosobenzene. In my travels, I've had some fun. Look at this sign, which I found in a European country, emergency telephone, 174 kilometers ahead. Well, if my car is broken down, I don't want to walk 174 kilometers for this one. And my concluding story really is, that invention of Viagra. So that was done for angina pectoris, but the innovators came to India. They were, of course, as every tourist does, going to the Taj Mahal, and they found this is a different cartoon, is valuable to learn from the mistakes. But how do we make a mistake without doing anything wrong? But this was the celebrated billboard. And look at it. It said, "Come via Agra, you will see man's greatest erection for a woman." Now they took a picture of this one. The man who posted this billboard sold 9 million copies of this T-shirt. Now, whether that is ingenuity or innovation is for you to decide. But this is the kind of fun I've actually had in chemistry. With that, I come to the end of this talk. I hope I've shared some perspectives for you. I hope I've succeeded in inspiring you, challenging you to a slightly different way of thinking. As students, all of you aim to assimilate a lot of knowledge, you absorb it. But now you should come to a stage in your life and career where you not only assimilate or absorb, but you leverage and vision. That should be a pathway going forward, and that will give you a lot of pleasure in life. I've been asked to give a talk to a broad general audience. I hope I haven't belabored the chemistry too much, but I hope I have also given you some food for thought, something to think about. Which direction are we going? There is a famous story told of a professor who had done this, and he was asked at the end of the day, the audience was asked, did the speaker succeed in conveying the enthusiasm? And this woman said, at the beginning of the talk, there were two people who knew what he was talking about. One was he, the other was God. At the end, this number has been reduced by at least one. I hope that did not happen here today. Thank you very much for your most gracious invitation, for your very kind hospitality, and for your patience in listening to my thoughts. Thank you.
Thank you very much, sir, for such a nice talk. Well, I don't know very much chemistry, but just I'm curious about one thing, that you work on like lots of bioextract. So the bioextract usually contains varieties of biomolecules, right? So how you usually identify them or separate them to use in your pharmaceutical?
The most traditional methods are to separate them by HPLC, which is high performance liquid chromatography. Then you purify these molecules. Then you get the complete range of structural information, including, if possible, the X-ray data. But UV, IR, NMR, and all that is absolutely very, very important. And then you can make valuable conclusions. Each plant will typically give us about anywhere from 7 to 9 molecules. Now, I am not an advocate of administering 7 tablets or 9 capsules to people. That's not the idea. But I want to study the chemistry and I'll prove it is non-toxic and then go back to administering the extracts. But now this will be with a firm scientific foundation.
Okay, thank you, sir.
I'll be delighted to answer any and all questions.
Yeah, audience, please ask if you have any queries.
Yes, Professor Akhilesh.
Well, that was a delightful talk. And I think you have missed your real career as a stand-up comedian. But I want to ask you something here. And that is, I think the question that I'm asking is something that probably Indian audiences will ask. So when you look at Indian pharmacopoeia, basically Ayurveda, that's what I'm talking about here. Okay. There is, as you said, there is a collection of some 300 odd medicinal plants, and that are administered for various, or their leaves or roots or whatever are administered for various ailments. So these must have, of course, been discovered by at least in the beginning, just by observation. People must have noticed, hey, you know what, you eat the leaves of that tree and your tummy doesn't hurt anymore, for example. Something of that kind must have happened. But reliance on that way of that kind of deduction is certainly not very scientific. Although in the beginning it is useful. In the beginning it is useful because you don't have anything else. Okay. So what should interested people do in order to make these studies or similar studies scientific? What's a practical way of doing these things?
The most
Not simply going to a Hakim, of course.
No. You don't go to a Hakim, but you take your top 10 medicinal plants.
Mhm.
And we all know what they are. In India is Tulsi, we have Ashwagandha. Many of these are sold as various formulations in the USA. But nothing is known about the chemistry. So what you do is you purify these compounds. You purify these compounds down to a pure structural form. Then you do the molecular biology. You find out what targets it binds to. Now you have some validated scientific data, but don't stop there. Do a limited toxicology test, just tweak it a little bit, and then find out if these materials are indeed going to be good for you. Now, with this scientific and validated data, you marry that to the clinical observations. You go to these doctors, they have got written down years of practice, which patients have been cured and everything else. Now, within a couple of years, you can compile a wonderful set of dossiers, which will convince anybody that you have some firm scientific data.
And so this is what perhaps ethnobiology or ethnobotany would be called, right?
Absolutely. Also, epidemiology, some of it will go into that one. Reverse pharmacology will go into it, and that's the team we have to assemble.
So once such a compound has been isolated, what are the next steps? How do you scale up and how do you commercialize? I think many of our students in IIT BHU would be interested in learning that.
No, exactly. So once you have done the structure proofs of some of these compounds and studied the biology, I'm not a big fan of doing different medicinal chemistry and the methyl ethyl butyl. You make different formulations. You make the nano and everything else. You can make the zinc nano, and again, you can do some very limited studies. Today, there's a huge market for that even in the Western world, literally. Many people are very excited about herbal products, but they are interested in getting, again, this scientific foundation. I'm working with a couple of companies. That is the purpose of my visit here. That let us put, again, this chemistry and biology, let us make some new formulations, but let's subject that to some rigorous studies. And I think the people in BHU and all can play a tremendous role. I envision there are student projects that can be run. I am visiting the Maharashtra University of Health Sciences, which is the current vice chancellor, is a very prominent physician who is very interested in doing this. So we need to put together a cadre of people who will study all this and thereby publish some very credible, internationally respected data. And
well, you are right about BHU, because BHU does have, besides having an All India, what do you call it, AIIMS, All India Institute of Medical Sciences, it actually for a very long time has had an institute of traditional medicines.
Yes.
I'm not sure exactly what the exact name is, but even when I was a student there, that was in the 70s, it was a big thing. And it's actually an ideal place for this kind of research to be conducted. Also, I think in the city, there are many practitioners available and who have, and since many of these practitioners have had information handed down from father to son kind of stuff, okay, they have old manuscripts. So this should be probably done on a very scientific basis, and BHU should take the lead for that.
I cannot agree. I cannot disagree with a single word that you have said. I'm a strong proponent of that one. The one thing that has inhibited this whole field is, as I said, everybody practices in a different language. And so far, there has been no unifying write-ups. In allopathic medicine, we have had that. Everybody learned in the medical college in English, everything else. Whereas the Ayurveda has been all over the place. I mean, whether you're practicing in Marathi, Tamil, Hindi, any one of a number of languages, the people are very intelligent, but there is no unification in this. And also what is being discussed in India right now under the new educational policy is that in America, we have a cadre of physicians who are MD, PhDs, which means they understand the medical research and they understand what it takes to practice medicine itself. In India, we do not have this cadre of people. So now they're trying to set it up. So these are physicians who will invest a certain amount of time in laboratory research.
That I completely agree with you. I think, as I was saying earlier, the disciplinary silos in India are very tall.
Absolutely.
And that needs to be dismantled, or at least, the walls have to be brought down substantially. Yeah. Well, come on, guys. Neela, Raje, Nikhil.
I noticed a hand.
Shweta.
Yes, sir. Hello.
Yeah, please go ahead.
Thank you, sir. Such a nice talk. Sir, I want to ask that if extract or some compound after synthesis process comes out to be toxic or show some side effects, then how it is treated in effective manner and what precautions should be taken?
The first thing, if it truly comes out toxic, you find out at what concentration, what dose has been administered. And normally there is a therapeutic dose and there's a very narrow window. But what you have to study is always study how much of the extract is being administered to the patient. And if it still all is toxic, then you just abandon that treatment. I'm not an advocate of endangering anybody by administering something that shows any toxicity. Now, one thing we are doing to de-risk this is we are working on the top X number of plants or other things which have been used for centuries and nobody has reported any real bad side effects for that one.
Thank you, sir.
The questions. Okay. Well, we will, as I said, Mukund, we will be posting this on the website on YouTube, and so this will be available, and I'll send you a recording too. And
please do that, and I'm just so delighted and literally honored that I was invited to do this.
no, pleasure is all ours, because, and not simply the pleasure, but a privilege too, because you have addressed these students. And what I always think is that even in the listeners, even if a little tiny seed has been sown, you never know when the tree will grow.
Absolutely.
Yes. Thank you.
Yeah. Thank you. Thank you very much, sir.
Well, bye-bye.
Bye-bye. On that note, shall we leave or?
Yes, I think so. Bye-bye.
But thank you.
Please. Bye-bye.
Bye.